Okay. Hi, everyone. So I'm Alice Lacan. I will introduce to you our survey paper on GAN-based data augmentation for transcriptomics. So first, a little bit of context. This work falls under the scope of personalized medicine. So basically, we use gene expression data from patients to predict some cancer phenotypes. In the literature, most classic machine learning approaches have been used to that effect. But considering the growing amount of transcriptomic data and the complexity of such data, it's only fair to wonder whether we could use more advanced deep learning methods to really harness the full predictive power of such data. The problem is that deep learning requires a big amount of training data, right? And gene expression data is scarce for different reasons. It's still expensive to produce. There's a lack of integration of different data sets. Also, depending on the condition you're interested in, sometimes you only have a few patients to sample from. And in addition to that, for each patient, you end up with a very high dimensional sample, right? From 10,000 to 50,000 variables. So you end up with a low number of samples that are high dimensional, and this can lead to different overfitting issues. And basically, your deep learning model will not be able to generalize well. So this is why in the literature, only a few studies have worked on bulk RNA data using simple autoencoders for dimensionality reductions or pre-training, for instance, and simple multi-layer perceptrons architectures for predictions. So it's tricky to develop more advanced architectures that could be more suited for this data. So the solution we propose in the paper is using data augmentation. So data augmentation is when you artificially enhance the number of training samples in your training set. It was first popularized in computer vision, where people started to apply geometric transformations or just colorization on the data to create additional samples. Also, in natural language processing, you can apply similar transformations like random deletion of words or synonym replacement. Now, you know that the butterfly here was still a butterfly, even though it was upside down, right? Because you have some domain knowledge about this kind of image. And similarly here, you know that the sentence keeps its overall meaning because you have some domain knowledge about this text. Now, it means you can apply transformations that are label invariant. The problem is that for gene expression data, we don't know what kind of simple transformations we could apply. A more proper approach would be to focus on model-based augmentation, where basically we use deep learning generative models to learn the distribution of the data and then generate additional samples. So generative models that can be mentioned are variational autoencoders, generative adversarial networks, GANs. Here are two examples of GAN-generated images. And more recently, the whole family of diffusion models have shown very impressive results, outperforming GANs in both diversity and realisms. Here are two examples of images that were generated by the DALI model. Now, even though we saw a lot of advances in computer vision, it's still hard to generate high-dimensional images. And considering the dimensionality of our data, it is a challenge to learn such a high-dimensional distribution. So it's a challenge that we mentioned in the paper. And also, for instance, here, if you look at this image, you can see that there's something weird going on with the hand, right? The fingers seem cropped, and you see some artifacts. So you know that because you have some domain knowledge about what a realistic hand should look like. Now, once again, for transcriptomic data, we don't have this overall insight. So assessing the quality of the generated data is also a challenge that we try to tackle in this survey. A solution that we suggest is bringing domain knowledge that we have on genes relationships, for instance. So in the survey, we investigated different generative models, and we focused on generative adversarial networks, GANs. So just as a brief reminder, in a GAN framework, you have two opposite teams. You have the generator and the discriminator. So the generator will take as input a random variable from a random distribution, like a Gaussian distribution, for instance, and it will output a fake sample. Now, on the other hand, the discriminator needs to distinguish between a true sample and a fake sample, while the generator needs to fool the discriminator into thinking that this fake sample is actually a real one. So you end up with this kind of min-max optimization problem that is illustrated by this loss function here. Now we want to help a little bit the generation process, so we condition both networks using clinical patients from the data, clinical data from the patient, sorry, like age, sex, tissue type from the sample. And this first conditional GAN is the first generative model that we investigated. Now, in practice, this has been shown to be very unstable to train, leading to convergence issues and mode collapse. So a lot of literature has been published on trying to make GANs more stable. And currently, the state of the art is the Wasserstein GAN with gradient penalty. And so we have the same framework, but here the discriminator becomes a critic. It does not output a binary probability anymore of a sample being true or fake, but rather a kind of realism score that it wants to maximize for real data and minimize for fake data. So we end up with this slightly different loss function here, in which we add a penalty on the gradient to regularize. So this WGAN-GP is the second model that we investigated. Now we wanted to boost a little bit more the generation process using attention mechanisms. So attention was popularized with transformers for natural language applications, and they have been shown to really boost deep learning architectures, especially allowing you to learn more dynamical relationships in the inputs. So here is a famous attention map of a French and English sentence. And you can see how the model learned how each word needs to attend to all the other words in the translated sentence. So we wanted to do the same thing, not with words, but with genes. So we added a self-attention module at the end of the generator here to really learn the genes' dependencies to others. Now you see that if we try to compute the attention pairs between all the genes and every other gene, as is done here, it becomes quickly impossible to compute, right? Because we're handling thousands and thousands of genes. So we needed to constrain the number of attention pairs we could compute. And to do that, we decided to include the domain knowledge that we had on genes co-expression and protein interactions. So basically, we keep only a certain amount of those interactions, and we built a knowledge graph that is illustrated by this matrix here that is quite sparse, right? So you will constrain the model to learn only specific attention pairs. And so in the original framework of attention, you would learn the similarity between one gene and all the other genes. Here, we really want to learn the similarity between one gene and a subset of genes it's the most correlated with or interacting with. So this will really force the generator to generate data with specific attention on some interesting pairs of genes. So like I mentioned, also one of the challenges is to assess the quality of the generated data. In the survey so we used data from the public Cancer Genome Atlas, so TCGA, holding roughly 10,000 of samples, bulk RNA sequencing samples, and 20,000 of genes. So in the paper, we gather different data quality indicators that can be used. First here, I mentioned the supervised indicators that require an additional predictive task. The first one we mentioned is the reverse validation, which is basically when you use your fake data, your generated data to train a classifier, and then you just compare the prediction metrics. And since the whole purpose was to show the gain in data augmentation, we also performed experiments where we trained a model using real data and augmented with generated data, and we compared also the prediction metrics. Now for the unsupervised data quality indicators, here I'll just go briefly over some really state-of-the-art metrics that are currently used in generative modeling. The first one is the Frechet distance score, which is basically the what's the time distance that you compute between a true and a fake feature vectors. Those feature vectors are usually obtained from a pre-trained model that would be suited for your prediction tasks. So here, cancer classification or tissue type classification, for instance. It gives you an idea of the similarity between your true and fake data in a more reduced space. And we also mentioned precision and recall. So the names are borrowed from the classification framework, but these are not the same metrics. Here, these are nearest neighbors based metrics. And precision here should be understood as how much precise, how much realistic is your data. And recall should be understood as how much diversity do you have in your generated data. We also mentioned the adversarial accuracy metric that was proposed in a paper for privacy preservation concerns. So now I'll just show you some of the results. First are here are some UMAP visualizations of the true data in the blue squares and fake data in the red squares. And here we notice that it seems that using a WGAN-GP with or without attention seems to preserve the tissue type clusters that are highlighted by the different colors here. But for more quantitative results, here are the results for the unsupervised indicators. And so here I present the best generative models that we obtained. And the main conclusion that we drew from this table is that the WGAN-GP without any attention was the best model that we obtained. It optimizes most of the metrics, even though it's important to note that there isn't one model that optimizes all of the metrics. And what's more interesting is when we look more closely at the precision and recall, we end up with a pretty high precision, right? 99% roughly, but a fairly low recall, around 50%. And it's interesting because it means that what we generate is realistic enough, but we're lacking a lot of diversity in the data. And it's really characteristic of GANs. It's more commonly known as the mode collapse issue, where you end up generating only subsamples of your distribution, so only certain modes, and then you end up lacking diversity. And what's interesting is that when we look at the different attention GANs with different training settings and graph knowledge that we use, you see that we're able to increase this diversity. Now, it's more visible here when we look at this scatter plot of precision versus recall. Here, the green point here is the result for the WGAN-GP without any attention. And you see that depending on the graph knowledge that you used or the training settings, in fact, you just slide over this kind of Pareto front here, increasing your recalls or increasing your diversity at the cost of your precision. Now, something that I forgot to mention now that I see this slide, so we also performed a lesion study for the attention part where we try to assess whether knowledge actually helped to build the knowledge graphs. So we also built random graphs with similar density. And here we see that actually there is no clear pattern whether using knowledge graphs or random graphs to constrain the attention part, whether it actually helped. So they work similarly. So we concluded that increasing the network expressivity, increasing the number of weights, actually could help the model, but the domain knowledge that we used to constrain the attention module didn't necessarily help more. Another way of looking at this diversity aspect is when you perform a principal component analysis. Here you see the PCA on here is the cumulative variance on the real data in darker blue here. And you see that when you look at the data generated by the WGAN-GP, so our best generative model, this red curve here, you see that actually most of the variance is explained by less than 500 principal components, meaning that, well, you have a big loss of information, right? You, once again, don't have a lot of diversity. And what's interesting is that this light blue curve here highlights the result for the attention GAN, meaning that we seem to preserve more information and diversity in this attention GAN generated data. So this echoes to what I just said earlier. And finally, so the results for the data augmentation. Here I'm showing results for 2 predictive tasks, cancer, binary cancer classification, and tissue type classification. Here on the x-axis, you have the increasing number of true training sample, and the baseline without using any attention is the black curve here and here. So you can see that, in fact, when we look at low sample regions, when we have less than 500 or 100 training samples, we can see that we have a significant gain in accuracy, right? Because here, for instance, we jump from 94% to roughly 98, and here 70% to roughly 94% of accuracy. Whether you use the WGAN-GP without or with attention. So there is a clear gain using data augmentation here. So just what are the main takeaways from this survey? First, if you use generative models to generate artificial transcriptomic data, you need a combination of quality indicators. Also, so what we concluded is that the WGAN-GP was the best generative model so far. Also, a surprising result was that, in fact, constraining attention using knowledge, domain knowledge, didn't work better than using just random graphs. And finally, like I just said, we showed that using data augmentation could really help boost performances, especially if you're in a limited, if you have limited number of training samples. So that's it for me. Thank you very much, and feel free to check the paper.